Good afternoon. Thank you for the opportunity to give you an update on the Omicron variant. I'm going to speak today about its genetic makeup and spread and try to catch some of the recent data, though this is an incredibly fast-moving field. I'm recording this talk 24 hours before you're hearing it, and in that 24 hours, it's possible that more data will have emerged, but let me cover what is currently available. I want to start off by reminding you that vaccine distribution is highly unequal. This is a schematic from the New York Times, and the darker the green in the color here, the greater the proportion of people who've received at least one COVID vaccine. And you can see that, for example, Australia, mainland China, much of the US and South America is a fairly dark shade of green, indicating good vaccine coverage. But in contrast, as you can see, much of the African continent is colored in very light green, and that indicates the fact that there's very little vaccine coverage in many countries across Africa. That is true of South Africa, where I'm based, and where, along with Botswana, the Omicron variant was discovered. And I just want to set the scene and explain a little bit about where we are in South Africa and what the context is for the emergence of Omicron in our country. On the left-hand side is another schematic that shows you the proportion of people who've been vaccinated, and South Africa is indicated there with the little red arrow. And you can see that our vaccine coverage in South Africa is not great, with less than 30% odd of the population having been vaccinated either partially or fully with a COVID-19 vaccine. Although our vaccine coverage, however, is relatively low, our seroprevalence is incredibly high, and that's shown on the right-hand side of the slide. And you can see various studies, which are listed down at the bottom, 5 different seroprevalence studies that have been conducted, with those data partitioned by age. And what you can see from each of these little graphs, as you look across the duration of the pandemic, moving towards the right-hand side, you can see that particularly, for example, in those individuals aged 35 to 59, the seroprevalence is in excess of 80%. So while we might have relatively low vaccine coverage, we have very high seroprevalence in South Africa, which makes it a slightly unusual situation. This is what our epidemic has looked like in South Africa. This is where we currently are in terms of new infections. We, like many other countries, we've had 3 waves, and I'll get into the genetics of those waves in due course. We are currently entering a resurgence for what looks like the 4th wave, and that wave is being driven almost entirely by Omicron, which I'm going to speak about in much of the rest of the talk. You can see in South Africa now, we are averaging over 15,000 new cases every day, and so really quite a dramatic resurgence occurring in South Africa at the moment. When you break that down by province in South Africa, you can see that many of the new infections that are occurring in this resurgence are in Gauteng, which is where Pretoria and Johannesburg are based in South Africa, and where the Omicron variant is really dominating infections. Infections in Gauteng account for roughly half of the infections that are currently occurring in South Africa. And many of them were early attributed to the Omicron variant based on spike gene target failure. And I'll explain a little bit more shortly why spike gene target failure is a useful finding that has come out of the Omicron emergence. In South Africa, we've been incredibly fortunate in that we've been able to rely on the Network for Genomic Surveillance in South Africa, which is a large network of labs across South Africa that have really done a remarkable job of defining at the variant level which variants are responsible for each of the waves in South Africa. It was leveraged off a group of labs that were funded initially through the National Health Laboratory Services. And you can see from that map at the top right-hand side that it includes many role players across South Africa. They've generated this network in total nearly 25,000 genomes since the beginning of the SARS-CoV-2 pandemic. And so we've been able to define really in good detail what the various waves have looked like. Our initial wave was the original D614G, that was followed by Beta, which many of you remember was incredibly neutralization resistant. Our 3rd wave was driven by Delta. We also did pick up at low levels a very mutated variant called C.1.2. It's extremely mutated and extremely antibody resistant, and we have described this in more detail in a preprint that's online. But I'm not really going to get into it, save to make the point that the C.1.2, while being highly mutated, is completely distinct from Omicron. Omicron, of course, is the focus of this talk, and it's responsible now for the 4th wave that's emerging in South Africa, and that's shown here in terms of percentage of genomes that are being picked up in South Africa in the red here. And you can see that with relatively limited sampling at the most recent time points as we fill in the gaps, but all the infections that are occurring now in South Africa can largely be attributed to Omicron. So what does Omicron look like? The Pango lineage name for Omicron is B.1.1.529. It's an incredibly mutated variant. It is the most divergent variant that we've seen to date. And you can see that on this phylogenetic tree over here. The phylogenetic tree, essentially how you read these kinds of circular phylogenetic trees is that the further a variant has gone from the center, the more mutated it has become. And if you can follow my line out, sorry about that, if you follow the line out here to this little red cluster of variants here, this is the Omicron variant. And you can see it has moved further from the center of the phylogenetic tree than any of the other variants that we've seen thus far. It's an incredibly highly mutated variant. It has 45 to 52 amino acid changes, including several deletions across the entire genome. And you can see this here up on this map right at the top here. So in the colored boxes of the various parts of the spike protein, which from a vaccine effectiveness point of view is of major concern to us. But you can see in the gray that there are mutations outside of the spike in several mutations across the genome. So it has picked up a really extraordinary number of mutations. As I mentioned, almost all of these spikes, or the concentration certainly of these, somewhere between 26 and 32 of these mutations occur within the spike gene. 15 of them occur within the receptor binding domain. And so really quite substantial mutations. The mutations include a deletion at position 69 to 70, which we are familiar with, having seen this before in the alpha variant of concern. And that is what results in S gene target failure. So what S gene target failure is, for those who are not clear on this, is essentially the failure of one particular PCR in a diagnostic test that relies on multiple PCRs. In the first wave in the UK, the alpha variant was tracked largely by the fact that there was an S gene target failure. And it's a very useful epidemiological tool because it means that we can get data fairly rapidly without full genome sequencing. In South Africa, it was really the S gene target failure that enabled the identification of this Omicron variant, because one of the local diagnostic laboratories in South Africa noticed that they were picking up a high level of S gene target failures. And so it does provide us with a useful tool, as I say, to track the epidemic, and I'll speak more about that in the next few slides. This is a slightly biased tree. It's South African oriented, and in this particular type of tree, the amount to which mutations have been accumulated is reflected by the move of the tree towards the right-hand side. And I just really want to make one point on this tree, which is, as I said, South African biased. It contains more South African sequences than global sequences. But just to make the point that this new lineage Omicron, shown here in red, is distinct from other variants of concern or variants of interest or variants under monitoring, including C.1.2, which is the highly mutated variant that we've previously described. So a very distinct variant that stands alone in the phylogenetic tree. Globally, the Omicron variant is far from limited now just to South Africa. This is a map that shows you the global detections of Omicron now across the world. So here, the darker the color, darker red indicates that more than 50 Omicron genomes have been picked up. And you can see certainly there is this very, very red area here down in South Africa where much of the sequencing has been done. But Omicron's now been detected globally in, as of today, 62 countries with more than 2,700 sequences across the world. So certainly no longer limited to Africa. And I think really speaks volumes to the fact that early lockdowns that were put in place to limit this were really far too late and not very effective, and perhaps caused more harm than good in terms of scientific research. This is looking now per country and looking to see what is known about the global detections of Omicron in those countries. So each of these bars represents a country. The first column here is the UK and the second is South Africa. And the colors indicate what is known about the particular Omicron sequences that were picked up there. Blue indicates a history of travel from South Africa or from a region known to have Omicron circulating, for example, Botswana. Orange are sequences that were pulled from samples that were identified as being potentially Omicron based on S gene target failure or similar. But I really wanted to point out in green, this is routine community surveillance, indicating that the likelihood that there is already community spread going on in several of these countries, and that Omicron may be much more prevalent than we have had initially suspected. This is a schematic of the Omicron spike compared to beta on the left and delta on the right. And I think this is a really beautiful way of understanding precisely how divergent the Omicron spike is from any of the other spikes that we've seen before. This is from the COG UK group. So in this case, each of these are a spike, and each of the red numbers here indicates a mutation away from the original ancestral virus. So for example, if we look at beta, we're used to seeing mutations at position 417, 484, and 501. This is the receptor binding domain here. And we're also used to seeing mutations here in the N-terminal domain. If you look at delta, again, very similar location of mutations. But look what you see when you compare the spike of Omicron to either of those things. It's a massive accumulation of mutations, many of them, again, in the receptor binding domain and the N-terminal domain, but really spread much more generally across the entire genome. And so when the sequences emerged that showed this level of extraordinary variation, it was a matter of 4 days, I believe, from the first description of the Omicron sequence to the World Health Organization designating this as a variant of concern, partly because of the massive number of mutations, but also because of the fast spread, which I've already alluded to. This is a schematic that drills down a little bit into the potential impact of Omicron spike mutations on transmissibility and immune evasion. If you go to the far right-hand side, this is another schematic of the spike. In this case, showing you more clearly where the mutations are that define Omicron. So each of these little red balls here indicates a mutation away from the Wuhan spike. And you can see that the mutations are largely within the receptor binding domain, within the N-terminal domain, and both the RBD and the NTD are major immune hotspots. But you'll see there's a substantial number of mutations here in S2. And that is unusual and has led to some concerns. If you look now at the schematic on the left-hand side, the red bar up at the top defines the mutations that are present in Omicron. So these are the lineage-defining mutations. Mutations that are shown in dark red are present in more than 50% of the Omicron sequences that we have. But there is, importantly, some diversity within this lineage. And in fact, literally every hour, more is coming out in terms of sequencing about the diversity that does exist within this lineage. And we can tackle that in questions. In the lighter pink colors, for example, this NKS here, are mutations that are present in some Omicron sequences, but not in all, somewhere between 5 and 50%. And then you can see that there are mutations that are rarely picked up in the Omicron. If you compare the mutations up at the top here that are shown in red with mutations that define other variants of concern or variants of interest, or in the case of C12, a variant under monitoring, you can see that some of the mutations that define Omicron, for example, here at position 484, are regions or sites that are frequently mutated in variants of concern. So for example, 484 is almost always mutated in a variant of interest or a variant of concern, and is again in Omicron, though in this case it's a E484A mutation, unlike the lysine, which commonly emerges at the site. I guess the point I wanted to make here is that there are clearly signals of immune evasion, but Omicron has a very, very different mutational profile compared to any of the variants of concern that have been described thus far. If you focus now down at the bottom here on the colors that have been used to highlight the spike mutations, they've been partitioned into 3 groups. So those that are shown in red, which are known, which are present in Omicron, but which have an unknown impact thus far. These are not mutations that have been defined in any other variant of concern. And so at this stage, much less is known about the role of these particular spike mutations. In blue are spike mutations, particularly in the NTD and the RBD, that we know from previous studies, either of variants of concern or of selection studies in vitro, for example. We know that these are known escape mutations. And then the other group of mutations I really want to highlight here are those shown in green here, positions 501, 614, 655, 679, 681, and 701. And these are mutations that are associated with increased infectivity or transmissibility. So really quite a large range of mutations here with a strong likelihood, and that has been confirmed in the last few days, that these will contribute to immune evasion. Then beyond spike, I just wanted to mention that Omicron also contains mutations at position 203 and 204, which are also known to impact infectivity, at least in vitro studies in cell lines. Right, let's now turn to look at the impact of these mutations. If I had given this talk 4 days ago, there would have been no data on this at all. And in the last 4 days, there's been a deluge of data, and we and others continue to produce data. And I think in the next couple of weeks, we really will have a sound understanding of precisely how much Omicron is able to evade antibodies triggered either by vaccination or infection. But I'll take you through what we have currently. This was probably the one of the first two studies that came out looking at the sensitivity of Omicron to antibodies. On the left-hand side is a comparison. This is data from Alex Segal and Sandila Tele in South Africa at RE. And what they did here was to compare D614G, which is the ancestral variant, with Omicron. In green are individuals who were both vaccinated and infected and therefore have very high antibody titers. And in orange are people who received only a Pfizer vaccination series and have slightly lower responses. But what you can see immediately is that when you compare the neutralization of D614G with Omicron, there is a 20-fold reduction in neutralization titers. So really quite substantial. To put this into context is a comparison here in the same model of D614G with beta, which we know to be neutralization resistant. And here you only see approximately a 5-fold reduction in titers. So this was one of the first studies that came out, which suggested that Omicron is very antibody evasive. At almost exactly the same time, a study from Danny Schurd and Ben Murrell at the Karolinska Institute showed very much the same thing, though the fold reduction here is slightly less. In this particular case, BD is blood donors and HW is hospital workers. And in both sets, the neutralization titers were assessed against the wild type and then against Omicron. And whether you're looking at the blood donors or the healthcare workers, you can see that there's a substantial reduction in neutralization of Omicron compared to the wild type. And when you plot that fold reduction, you can see that it's somewhere around about sevenfold, so slightly less than had been reported in the live virus neutralization assay. This, I should say, is a pseudotyped neutralization assay. This is data from our lab, also now comparing people, and I'm sorry, we've lost the legends at the bottom here, but it's shown here. So in black is the wild type, in blue is neutralization of beta, and in red of Omicron. This is looking at sera from Pfizer vaccinees, which have relatively high titers, somewhere between 100 and 10,000. But you can see a massive drop-off against beta, which we previously reported, and an even more dramatic drop-off against Omicron, with very, very few samples showing residual neutralization of Omicron in our hands. Even more dramatic with the Johnson & Johnson vaccine in South Africa. These are the two vaccines that are used in South Africa. But you can see here a drop against beta and an even further drop, in fact, no neutralization detectable in our hands from the J&J vaccinees against Omicron. Again, substantial drop in neutralization. This is a fourth study. This is data from the Rockefeller group looking at the effect of Omicron mutations on individuals, again, comparing Pfizer vaccination with Johnson & Johnson vaccination. In these graphs, each of the dots represents a plasma donor. In gray here is the wild type, Wuhan 1, and in blue is Omicron. In red is a sort of a hybrid spike, SARS-CoV-2 spike, that contains many of the same mutations that define Omicron, and you'll see that the patterns are very similar. If you look here at Pfizer vaccinees 1 month or 5 months post-vaccination, or at Johnson & Johnson vaccinees 1 month or 6 months post-vaccination, you can see that compared to the wild type in gray, there's a huge loss in neutralization against Omicron. So while overall Pfizer vaccinees do have some activity against Omicron at 1 and 5 months after the second shot, only in this assay about 50% of the Johnson & Johnson vaccinees had activity at 6 months after their first shot. The place where we do seem to see residual activity, and I alluded to this when I showed you the data from Alex Segal's group, where those individuals who've been vaccinated and infected showed increased neutralization. This is a similar analysis from the Rockefeller group, looking at people who were infected and vaccinated. And you can see here, in all cases, activity is retained against Omicron. So vaccination after infection certainly boosts the antibody response to very high levels. And I guess that seems to be the take home from all of these neutralization studies that have come out so far, is that if you start with an extremely high titer against the wild type Wuhan 1 or D614G, then you are likely to retain activity against Omicron. This is data asking what happens after a third Pfizer vaccine. So this is 1 month, 5 months, and then a third boost. And you can see massively high levels of neutralization here against Wuhan 1, with lots of activity retained against Omicron. So it seems that although sensitivity to neutralizing antibodies triggered by only 2 doses of J&J or 2 doses of Pfizer, many of those individuals show undetectable neutralization. A third boost takes you back into the territory where Omicron would be neutralized. And so that's where I'll end, is that the emergence and rapid global spread of Omicron, which is extremely antibody resistant and appears to be highly transmissible. I haven't really gotten into that, but certainly seems to be very transmissible and is accounting for all the infections in South Africa currently, really highlights the incredibly important role of rapid genomic surveillance linked to quick immune profiling to understand this. And in the case of Omicron, it seems that the reduced antibody titers may result in the decreased ability of vaccines, all the vaccines potentially, to prevent infection. But I think it's really important to point out that prevention of severe disease is likely to be retained. And this is data that is based on in silico analyses suggesting that T-cell epitopes are very likely to be preserved in Omicron. We really have to wait on this for effectiveness studies, which are ongoing in many, many countries as Omicron infections occur. And I'm going to stop there and thank the Network for Genomic Surveillance in South Africa. This is a huge network that involves many people, and I'm just quickly flashing through this, but to acknowledge the vast effort that they have made and the rapidity with which they've shared their data, and then to thank our lab. And I'll stop there. Thank you.